Well, thank you very much for inviting me to speak. Somebody was just saying that there's a known inverse correlation in between how much you enjoy a conference and how late on in the conference you speak, and I'm sure that's true. And it's a testament to this conference that I've enjoyed it tremendously nonetheless, so thank you. So as Ollie mentioned, I'm moving in 3 weeks to be to a building close to this building. This building now looks about 90% like this one. In fact, it looks very much like that on the outside. It doesn't look like it on the inside. New Big Data Institute, which is part of the Departments of Medicine and of the Department of Public Health at Oxford, which is really going to look at many issues in big data related to health, including infections. And we're actively recruiting in infections. So for people who want to set themselves up as PIs, and I'm recruiting within my group as well, particularly in viral phylogenetics. So I take the opportunity to say that now. And the director will be, is Gil McBean. So I want to take a slightly public health perspective, and I want to try and zoom in on one particular topic in HIV public health. So you've all seen this graph or graphs like it, which is a spectacular success of antiretroviral therapy. The UNAIDS estimating that globally something like 30 to 40% of HIV-infected people are now on antiretroviral therapy. And in blue, you can see the number of AIDS deaths having started to decline down to something like 1 million AIDS deaths. So still very far from defeated, but substantial and sustained declines in AIDS mortality. In the last few years, this progress, which has essentially been about treatment, has been allied with prevention, and really a galvanizing finding in this long-standing idea that treatment, by reducing viral replication and also viral shedding, can also block transmission, was this paper, the findings of the HPTN-052 study, which were nominated as a Science Breakthrough of the Year. And the summary of the finding is that in couples who start where the HIV-positive person starts antiretroviral therapy early and where viral suppression is maintained, antiviral therapy has 96% with some uncertainty efficacy in blocking transmission. In fact, there were no phylogenetically confirmed transmission in those on treatment, apart from one couple where the transmission occurred on something like the first day after the start of antiretroviral therapy. This has led to a large movement. This is a declaration from the United Nations, a political declaration. There are very few political declarations by the United Nations to end the AIDS epidemic by 2030. It includes 20 fairly specific targets, which cover all of the important areas, really. Financing, which is the toughest area, how this might be done, focus on pediatric infection and adolescent girls and discrimination and stigma. No focus on finding undiagnosed men, which I think is very important. And might be of interest to many of you, also includes very ambitious targets for hepatitis B virus, hepatitis C virus, and tuberculosis. The specific aims of the declaration are to reduce in the short term by 2020, so by 4 years, the number of HIV infections to fewer than 500, the number of AIDS-related deaths to fewer than 500 per year, and eliminate HIV-related stigma and discrimination. So to put that in perspective, in 2015, the United Nations UNAIDS estimates that 2.1 million people were infected last year. So getting that number from 2.1 million to 500,000 is not trivial, whereas AIDS mortality is now down to 1.1 million. You'll notice, just from an ecological perspective, many more new infections still than people leaving one way or the other, either by starting treatment or dying from AIDS. So the HIV-infected population is growing very rapidly. Stigma is very widespread. You'll also have seen figures like this. I just want to point out one aspect of, well, actually, I want to point out two aspects of those figures. The numbers are spectacular in terms of the number of people on treatment. and the increases are spectacular. Nonetheless, the rate of increase has actually slowed slightly, and in all but one of these years, say in the last year, 2 million people were started on antiretroviral therapy, but 2.1 million people were infected. So there are still more people getting infected than starting antiretroviral therapy. So we're nowhere near the state of actually meeting the need for antiretroviral therapy. And the problem is still growing. So contrary to all of the news, HIV has become something of a good news story, and so it should be, because so much has been achieved. But it's not over yet. The other thing which hasn't really been emphasized, and I think it's time to start thinking about alarm bells, are these curves. So these are the number of new infections in the 6 WHO-designated zones. And you can see in all but one of the zones, the number of new infections per year is estimated to have been absolutely constant, despite this rollout of antiretroviral therapy, apart from Eastern Europe and Russia, where the number of new infections has been growing rapidly. Maybe the hint of an inflection here in sub-Saharan Africa. The y-axes are different in sub-Saharan Africa. There are some isolated good news stories. Maybe Uganda has been a good news story. Zimbabwe might be a good news story. But I think it's time to be concerned about this. Also note that these political declarations, unfortunately, don't always have the desired impact. In 2011, the last political declaration by the United Nations had declared that we should aim to reduce incidence by 50% by the end of 2015. And that clearly hasn't happened. One comment is population has been growing over this time. Global population is growing rapidly, so this is reduced per capita incidence. So in summary, there are spectacular successes in preventing progression to AIDS of deaths, and those should be celebrated. But there are disappointing reductions in HIV incidence, and the number of new infections is still outpacing the number of people starting antiretroviral therapy. So why is incidence not declining? Well, one possibility is that it is, but we're not measuring it correctly. So the way incidence and ART coverage is estimated is through surveys and extrapolating, and every few years there's a large correction in HIV statistics when people cross-validate the projections with large censuses, but the censuses typically only take place every 5 or 10 years, and those corrections can be large. I will note that the last census in South Africa actually concluded that there were more, that incidence was going up faster in South Africa than UNAIDS had projected. So that can work both ways. I've already mentioned population growth. It could be that treatment is very effective at blocking transmission, but that a lot of transmission is taking place before people start treatment, particularly concerned because of the very high viral loads during acute infection. It could be that there are still many undiagnosed people, especially men. All of the studies, really globally, if you just look at recruitment statistics in all studies, very typically, unless it's a specific study on men who have sex with men, all of the studies typically record twice as many women as men. It's just much harder to find men, and that's true in health services generally. It could be poor viral suppression. There are actually lots of cross-sectional surveys suggest that this isn't a problem, that many of these 17 million people now on ART are actually virally suppressed. In some areas of the world, it might be more risky behavior, or what's known as risk compensation, because of perceived reduced risk, people engaging in more risky sex, or it could be something else that we don't understand, or a combination of all of the above. The specific question that I want to ask in this talk and address is what can HIV phylogenetics add to this? And I'd suggest, uncontroversially here, that HIV phylogenetics provides insights into sources of transmission to help understand the past epidemic and therefore to improve prevention in the future. so many examples. I wanted to get to some details of our studies. There are many, many great studies out there, and I just decided in the interest of time not to review them. One simple but notable application of phylogenetics is that if the HPTN 052 study had been done without phylogenetics, the efficacy would have been 89% because of 4 seroconversions in the couples where antiretroviral therapy was taking place. But actually 3 of those were not transmissions within the couple. They were the negative person getting infected with somebody outside of the couples. And phylogenetics showed that only 1 of the transmissions was plausibly within the couple, resulting in much increased efficacy. So I want to begin by talking about our study in the Netherlands. Why do we work in the Netherlands? Well, it's partly a historical accident, but it's partly it's an amazing example of what can be done with internal data sharing. It's not open data because of issues of confidentiality, but it's the only country in the world, I think, where all clinical centers all share data centrally within a central database, in the sense that it's a true national cohort, which includes every single infected individuals, with an opt-out policy and with a tiny, less than 1% of individuals opting out. Drug resistance testing is done for almost all individuals, or it's done for all individuals now, and the poll sequences are now being shared in this national database and linked to all of the rich clinical data. So the full clinical and epidemiological history is available for each individual, and as expected, you get a wide distribution of subtypes, with many, the subtype B epidemic being dominated, but not exclusively, men who have sex with men, and many other subtypes associated with immigrants, representing about 40% of all infections. So some findings of a study led by Daniela Bezemer and Corinne in my group. We just looked at the distribution, the temporal distribution of new diagnoses in different clusters. So each line here, each dot is 1 individual, according to their date of diagnosis, and each line is 1 phylogenetic cluster. So there are 91 large clusters where the majority of people are MSM, men who have sex with men. And you can see two notable findings are that 60% of MSM clusters are old clusters, and the clusters never stop. There isn't a single case of a transmission chain actually being interrupted after the introduction of antiretroviral therapy. So not only did the epidemic not go into decline, none of the sub-epidemics did. Here are the heterosexuals, 1 very large injecting drug user cluster, which has become increasingly heterosexual, and many individuals who don't fall into clusters, who are very rarely described in clustering studies, here falling into small clusters or no clusters. Individuals are asked whether they think they were infected abroad, and if you don't fall into a cluster, validating the clustering to some extent, you're much more likely to self-report being infected abroad. One potentially piece of good news is that these clusters are aging, so you could think the epidemic might be aging away. So if you follow a cluster, individuals are getting older at diagnosis at about 0.5 years per year. However, these recent clusters are clusters of predominantly younger individuals. If you calculate the reproduction number for each of the clusters, each one of these lines is a reproduction number for 1 cluster, you can see that many of the clusters have very similar reproduction numbers, except the most recent clusters shown in green and yellow, which have higher reproductive numbers, and those are the clusters amongst younger individuals. To try and understand the dynamics a bit more closely, we tried to identify possible transmission pairs. This is very much not proven transmission pairs. This is putative transmission pairs. And the possible transmission pairs were identified just by pol similarity in the pol genes, patristic distance. The patristic distance was adjusted for the amount of time since infection. The recipient, we typically knew when the recipient was infected, and then we identified their possible donors. And the question was, what clinical stage were the possible donors in? So here are all the 13 different clinical stages we looked at, including recent infection all the way to viral load suppressed. And the conclusion was that the overwhelming majority of transmissions came from individuals who were recently infected, undiagnosed. And 43, this was sort of additional modeling, 43% of all transmissions occurred from people who were in early HIV. So in terms of antiretroviral therapy, it seems like within the Netherlands, pretty much the potential of antiretroviral therapy to block transmission has been largely reached. Retention in care is exceptional, suppression is exceptional, and actually treatment seems to be quite permissive of blips in viral load and so on without much transmission taking place. One of the things we then did is we remodeled on top of the epidemic what might have happened if different, because we had the full clinical history of individuals, we asked the question, well, what would have happened if individuals were in different stages? What would have changed? And we remodeled. These are, you can't see all of these are lots of different scenarios. And because we had the testing history of individuals, we knew when individuals had previously tested, we found that lots of individuals who seroconverted had actually relatively recently had a test where they were found to be negative. On the other hand, people who become transmitters test much less in the population on average. So we suggested that pre-exposure prophylaxis, which is antiretroviral therapy for people who are not infected in order to stop them getting infected, could be targeted at people who voluntarily come in to test. So it seems like people have a fairly good idea of whether they're at increased risk, and if they're coming in for testing in a completely non-stigmatizing way, you can say, have you considered having pre-exposure prophylaxis? And that's been shown it can be taken daily, or it can be taken before you know that you're going to undergo some risk exposures. So next I want to talk about the Beehive study, which is a European-wide study. So this is a study that we've set up bringing together data from several cohorts across Europe. And actually the primary purpose of the study wasn't epidemiological. It was to address a hypothesis that I've been working on for many years now, that the virus plays an underappreciated role in terms of predicting viral load. So the conventional wisdom has been in the field for many years that viral load is predominantly determined by the host genetics and the host immune response, and based on a number of sort of circumstantial lines of evidence, but increasingly solid lines of evidence, we hypothesized that the viral genome, the actual viral strain, influenced viral load. And that motivated this study, which is finding, we've now found over 3,000 patients, and the study is aiming to get 4,000 of these seroconverters, where we get an early sample and we determine the whole viral genome, with the primary objective being a viral GWAS to find the viral determinants of severity and to do molecular epidemiology and to look at the clinical relevance of dual infections and minority variants. It's a wonderful collaboration that we've brought together for this, and it's been a real privilege to be part of this. This sequencing has been done here at the Sanger by Paul Callum and Astrid Gall, leading this in Paul's group, using a 4-amplicon approach. Opinions. The advantage of this is that it's very straightforward to do it with very large sample sizes. You target 4 conserved regions of the genome, which are conserved across subtypes, amplify the virus, and obtain high coverage of Illumina reads across the genome. So as most of you know, the problem of how to actually reconstruct what viruses, you have a diversity of viruses within the individual, and you end up sampling these diversity of viruses, chopping them up into bits, amplifying them, chopping them up into smaller bits and making your libraries, and then you end up with these small bits of sequences, which are actually never really 300 base pairs long. And the question of there are well-established or sort of well-established ways of reconstructing the consensus genome, so the genomic average of all of these viruses. But what more can you say? Well, the first thing we did, Chris in my group and colleagues here and many others did, was put together a pipeline to try to maximize the coverage we get in the consensus genome. And we found that the methods available involving de novo assemblies and mapping both had their drawbacks. Mapping induces a bias to the consensus, which is terrible for phylogenetics, by the way. It makes your virus look more like the consensus than the virus is, whereas de novo assemblies generate partial virus contigs, which are hard to interpret and include lots of contaminant and rubbish. So this Shiver pipeline is now a sort of one-line pipeline which generates a consensus genome and all of the minority alleles and a BAM file. And Chris has shown that this outperforms existing pipelines for doing this task. So once you get these consensus genomes, you can do assembly. You can do phylogenetics on the genomes, and compared to pol genes or other genes, what you really get is an increase in resolution. One of the nice things, you still, because of the recombination rate, you still don't get high resolution to the base of the phylogeny. But one of the things you see is you go just far enough to see that the, particularly in this case, the Dutch and the UK epidemics for these individuals, for this, what we sampled at least, was heavily intermixed. It was something that wasn't apparent with the more limited resolution of pol genes. Also, the other thing that happens is because you're averaging over many more sites, which are you end up with a much better molecular clock. So the R-squared, if you look at many of the pol studies or the M studies, you see R-squares of 10 or 20% are typical. But the root to tip distance as a function of time, basically the tree becomes much more clocky, and you see these very high molecular clocks. So I wanted this talk to be predominantly about public health, but a topic dear to my heart is virulence and heritability. So I do have one slide about heritability, which I'll try and describe in detail. So we published a series of papers, and others published. Heritability is a proportion in the phenotype which is explained by the genotype. So the classic heritability study is what proportion of adult height is explained by human genotype. And the method, which has been around for 100 years for how to measure that, is you compare parents to their offspring, and the slope of that regression tells you what proportion of height is attributable to genotype. And we applied the same method to HIV. We looked at transmitter-recipient pairs and inferred that 30% of heritability, 30% of variation in viral load was attributable to the viral genotype. That's surprising because the viral genotype is something unstable, which is rapidly changing within the host. It's also surprising because it's much higher than estimates of the host contribution to viral load. Then some other studies came out which used phylogenetic methods. So this was on African cohorts. In European cohorts, there are no studied transmission pairs. So in Europe, it was necessary to use a phylogenetic method, where essentially the question is, if two individuals are close to each other on the phylogeny, do they have a more similar viral load than if they're far away on the phylogeny? And these resulted in really widely ranging estimates from the Swiss cohort study. Samuel Alison, who was the first person to do this, publishing an estimate of 60% heritability, but actually with confidence intervals which included 0. We also essentially published a study showing range from 0 to 100%. And then with a much larger data set, Emma Hodcroft and Andrew Lee Brown in Edinburgh published an estimate with narrow confidence intervals saying that it was 8%. So much lower than our transmission pairs suggested. But this year, two groups in Switzerland, Gabriel Leventhal, Sebastian Bonhoeffer, and Benjamin Mithoff, who's here, have shown that the models which people have used to estimate heritability on phylogenies are not robust, they're these Brownian motion models. And when we repeat the Brownian motion models used for the Hodcroft paper on our data generated with these high-resolution phylogenies, we get estimates which are actually very similar to the Hodcroft et al. Estimates. But when, as done in these groups, we use an Ornstein-Uhlenbeck model, which allows for convergent selection on viral loads. So the viral loads are not just going to get more and drift to become more and more different over time. Then we get estimates which are actually identical. When we look at the set point viral load, we get estimates which are identical to the transmission pair estimates. And reanalyzing the Swiss data, this group here is actually found. So the estimates of 30% are now very stable and consistent between phylogenetic studies of subtype B in Europe and transmission pairs in sub-Saharan Africa. The one thing that we did was really based on a hunch, which was a very expensive choice, we were worried that different viral load assays, are developed by different companies, and there's a large literature standardizing these assays, but they fundamentally operate in different ways and generate noise. So we redid a large fraction of the viral loads. We actually redid almost all of the viral loads. And when we do that, this more robust form of heritability produces much higher estimates of 48% across all subtypes, or 56% within subtype B. So this suggests that more than half of that actually heritability was being depressed by the use, just the noise of having different viral load assays, and that when we redo the viral loads and use the most robust method, we're now up to the point of about 60% of variance in viral load is attributable to the viral genotype. And I want to address the question of transmission and paraphyly using NGS. So Annemie van Dam already mentioned this in the, or actually discussed this in the context of criminal transmission. So this is a paraphyletic clade. So if you have multiple viral strains from a particular patient, and you have multiple viral strains from another patient, you get much more information than if you have one consensus virus from one patient and one consensus virus from another patient. In particular, a paraphyletic clade, you can infer descendants. So you can infer ancestry. So in this case, you have very strong, whichever method you use, you strongly infer that the ancestor here is the ancestral population to A, and that the ancestor of B is descendant, is within the population within which A, and you can infer something about the direction of transmission from A to B. So in terms of epidemiology, that's incredibly powerful, because you can then you can identify people who are likely sources of infection and really look at them epidemiologically. So one of the big missing pieces of information in conventional epidemiological studies is that you can't infer who are transmitters, and that's really the key thing for prevention, is working out who are transmitters, where are they geographically, where are they clinically, what are things which help us to find people who become transmitters to make sure that they receive the prevention services on offer. So paraphyletic clade is a specific indicator of recent transmission. And it's worth knowing there are a few really important points. One, that you can't rule out intermediates, but for epidemiology, that may not matter that much. If what you're really interested is A a transmitter or is A not a transmitter? It matters, obviously, for legal case. It may be that you just end up, and I'll show this in a minute, in some cases you just end up with a phylogenetic mess. There may be these missing intermediates, and if a lot of time has elapsed and since a transmission event, these two clades will start to diverge from each other, and you'll just get two neighboring clades. And Ramirez-Severson has done some very nice simulations showing over how much time you might expect these clades to start diverging from each other. So to look at this and other questions, Chris has developed this phyloscanner pipeline. So it takes these consensus alignments with all of the minority variants, and we had a feeling that you could, reconstructing haplotypes from short reads is really hard, and maybe onto a hiding for nothing, because there's so much recombination within host, both real recombination and induced by the PCR process. That really what we want to know is what's the phylogenetic signal in regions of the genome, and that actually our reads might just be long enough in order to be able to say what the phylogeny is of short reads and infer the ancestry of this bit in the genome in a within and between host manner. So we take these consensus genomes and align them. It's not straightforward because of all of the indels and so on. And we take all of the short reads which span a window, and we make a tree, and then we compute a bunch of summary statistics for that window, and then we scan that window across the genome, and we look at how the trees change as we go across the genome, so the stability or the difference of information. And that means that while these windows are quite short, you end up with phylogenies of maybe 350, 400 base pairs long, which you don't have a lot of confidence in any given phylogeny. You can test whether you have confidence in the summary statistics that you build up as you scan across the genome. The algorithm has a few nice properties. It merges overlapping paired reads to make synthetic longer reads, which helps resolution. It merges for computational efficiency. It reduces the number of reads present and includes, in a tunable manner, you don't have to do this, but to make it go faster, and include only the most common reads. So we don't merge the reads to consensus, we just include the most common ones. And because these are quite short reads, convergent evolution or selection or something could easily mess up the phylogenetic signal. So it excises, positions, all the drug resistance mutations, and all of the known epitopes of CTL and so on from standard libraries. And again, that's just a file that you submit. It runs pretty fast. In a day, you can process all of the MySeq files from 700 patients, and it works okay with HiSeq files. Takes a little bit longer. And you end up with these big trees. You end up with an alignment for each window in the genome, and you end up with one big tree for every window in the genome like this. You can't see much, obviously, patients at the end of this. And what you can do is you can then you can do kind of coarse trees, and then you can identify groups of patients that might be interesting and zoom in and just repeat the procedure to increase your resolution. Oops, and I pressed the wrong button. So just zooming in to a few patients, here's a typical single infection. Here's another infection. So a typical single infection, you have an amount of diversity which represents how much diversity there is in that patient, just so that you can see properly. This is what you end up with, and you get the different reads. These are quite heavily cleaned reads. There's lots of cleaning that goes in the previous pipeline. And you can, each read is given an identifier. And the next step we're just starting on is merging these in a phylogenetically sensible way across trees. So you get a unique identifier for each read. And you can see that they vary a lot in their frequency. So most of the reads, 200 out of something like 200 of the 30 reads for this patient, are actually aligned with this tip here. In this case, this is clearly a dual infection, and the nice thing is you can quantify how many reads make up this infection and how many reads make up that infection, and you can see how that varies as you go across the infection. The other thing which is really important is that it identifies contamination. It identifies contamination if you've got the contaminator. And we see a lot of contamination. So one really important lesson about this is when people say there is contamination, there is contamination. And if you don't have all of the data that's been running on your sequencer, just be really wary of reads with low counts, because they're more likely to be contamination than anything else. So we can spot contamination, and we remove those before processing further. And we see things that look like potentially a donor-recipient clade. So this is a paraphyletic clade. To improve the resolution of the paraphyletic clade, what you do is when you get a potential donor-recipient pair like this, or and emphasizing again for legal purposes that there could be a missing intermediate, you zoom in by including all of the reads, and when you include all of the reads, you can see that it's very robust, and that it seems like this is, as is well known for HIV, this is a case of a bottleneck with a single ancestor to this B557 patient within the population of B804. The nice thing is that you can then concatenate that information across a genome. So here you can look in a different part of the genome. In this case, either because of more noise or more resolution, you have to then go back and do bootstraps and so on. It looks like there are actually multiple, the two populations in this window are multiply intertwined. You can see that the dual infection here is not so well resolved. And actually, that's another nice thing that pops out, is when you get the dual infections and you see a window that comes out funnily, it's almost certain that you've just hit on a window where recombination happened. And bang, when you go in this one, you see very clearly a recombination in that particular window for that patient. So you can just statistically pick out the windows where recombination takes place. And to improve the sort of phylogeneticness, all trees excise the drug resistance mutations and the CTL escape sites. The bootstraps, you may worry that all of these trees have incredibly poor resolution. The bootstraps for the important nodes look pretty good. This formalizes some of the approaches that several people have used on a case-by-case basis, including Ollie and Richard Neher, who's sitting here, and Anna Mee, who presented something like this in the case of court cases. So in summary of the pipeline, you scan the genomes in windows 350 base pairs long, steps of what you want. You prepare your within and between host alignments of paired reads, which is not so straightforward because of all of the indels. You remove the clear contaminants and flag possible contaminants, merge rare reads into common reads. You estimate the phylogeny, currently using RAXML, produce summary statistics to assess clade structure for each patient, identify dual infections, identify paraphyletic clades and transmission clusters. You then have summary statistics scanning across the genome. So this is a single infection where this is one summary statistic is how many clades do all of the reads make. In this case, this patient only makes one clade as you scan across a genome, and then you can take the mean of that. In this patient, you get lots of different clades, and if you look at the root to tip distance overall is massive, but the root to tip distance within any one of the clades is very small, so this is a dual infection. How to precisely identify dual infection we're still experimenting with, but the mismatch in between the root to tip distance overall and the root to tip distance within the largest clade works well, and you can then also quantify, just as a summary statistic, what proportion of reads are within your different clades. Coming back to the question of transmission, Matthew Hall, who's recently joined our group, is sort of summarizing the information across different trees and has presented a poster here describing this. So in some windows, you find that all of the reads are side by side. In some windows, you find that they're interlinked. In some windows, you find clear directional transmission. And you summarize that just as a relationship in between different patients. In this case, there seems to be a very clear evidence of directional transmission. Again, there could be a missing intermediate, but there's much more evidence for transmission occurring this way than that way. And then when you concur back with the dates, that matches exactly. One of the questions is, should we be using the dates in sort of inferring these trees rather than using the dates to check for consistency? So that then gives us transmission networks, which we can start to do epidemiology with and go right back to the question we started with, which is how do you learn the lessons you need to target prevention? Here are some national and multinational. This is a Dutch-Belgium cluster, perhaps. And you can see in most cases, we actually have very little evidence of any particular direction of transmission. So this is a really important lesson, is that if you had stuck to one or two windows of the genome, you might have inferred strongly the direction of transmission one way, but it's actually quite rare that we get a consistent message of directional transmission across the genome. So in summary of that, phylogenies of PED and Illumina reads provide enough resolution to resolve mixed infections. Search for contamination by comparing identical reads, but transmission is apparent as paraphyletic, but it's not unambiguous. Evidence builds up by comparing trees across the genome, and we're now building these phylotypes by merging the reads in a phylogenetically sensible way across the genome. Then in terms of the epidemiology, I want to describe some work which is sort of earlier in the process of generalizing some of these findings to much more diffuse but much bigger epidemics. The first is the Pangaea study. Pangaea study now involves 8 major groups working in Uganda, Botswana, South Africa, and Zambia, and sequencing taking place here at the Sanger Institute and at the Africa Center, led by Paul Kellam and Tulio de Oliveira. The PI of the whole study is Dinan Pillai, and Andrew Lee Brown and myself trying to coordinate analysis activities. And this will, once the data are properly brought together, this will be an open data initiative with suitable metadata. And if you want really rich metadata, then you'll have to collaborate with the individual groups. One of the things we did is we used a simulation that we happen to be, this is a very detailed individual-based simulation, which we're developing. There's a long story behind this simulation, but it's a simulation of transmission that we're doing in order to help interpret the trial. It's a simulation which has been under development for 3 years now. We're hoping to make it numerically efficient enough that you can actually fit to trial data and to phylogenetic data. So it produces epidemic curves and it produces phylogenies developed by Ann Corrie and Mike Pickles. We and the Edinburgh group produced some of these simulations, and we asked a wider range of phylogeneticists and phylodynamicists to try and infer epidemiological dynamics, so doing this phylodynamics. And these were the groups which were able, under the type timelines, funder requested timelines, were able to produce results. Phylogenetic inference, this was a blinded exercise. These were the final submissions. And here at the top, you can see, this is submitted, should come out soon. At the top, you can see the estimates of incidence, and at the bottom, the estimates of the reduction in incidence. You can see really reflecting the differences in maturity between the methods and the amount of customization and time that people put in operating the methods, differences between the different groups in terms of the both the accuracy and precision of the estimates of incidence and change in incidence, but the Cambridge, London group, Eric Volz and Hussein and Simon Frost, clearly produced the most accurate and precise estimates, showing that phylodynamically it is possible to infer incidence in a generalized epidemic simulation and to detect changes, recent changes in incidence. Finally, to this question, why is incidence not declining, and can we show whether it's possible to make incidence decline? I want to talk about a study that I've been involved in for a number of years, known as the HPTN 071 study. So it's within the same organization as the HPTN 052 study, also known by the acronym Popat, which stands for Population Use of Antiretroviral Therapy to Prevent Transmission. It's an NIH and Gates-funded trial sponsored with PIs Richard Hayes, Sarah Fidler, Helen Ailes, and Nolda Byers, operating in 21 very heavily HIV affected communities in Zambia and South Africa, where adult prevalence is actually 20 to 30% in many of these communities. The baseline ART coverage is much lower than UNAIDS suggest, and HIV incidence, we don't know HIV incidence, but we estimate it to be at least 1.5 or more percent. The intervention is, the trial involves 1.2 million people in communities which are randomized to 3 arms of the trial. The intervention involves hundreds of community workers going house to house and providing an intervention package. The intervention package consists of testing everybody in the household to find out, for them to find out whether they're HIV infected, and offers them immediate antiretroviral therapy if they're infected, circumcision if they're male and not infected, and a whole prevention package of counseling and it really drives home the amount of logistical work which has gone into this trial. Really, in fact, the trial has been criticized for being so big. Why did it need to be so big? But I think it's the only way to really find out what's involved in this intervention, which is being now promoted by the United Nations. And it's important to find out, given the sort of very mixed data from the UNAIDS, whether it really can, whether people want this intervention is the first question. Whether it can be delivered is another important question. And if it is delivered, does it reduce incidence? The second arm is treatment isn't offered to everybody, but is offered according to national guidelines. And arm C is basically just national standard of care. In a completely separate, again, huge effort, there are 21 cohorts of 2,000 adults who are being visited in 4 annual rounds to measure whether the infection rate was different or not. So we've got intervention teams and incidence teams. The question, so my team's involved in 2 activities. One is modeling, which has been part of the design of the trial, which is part of the ongoing evaluation of the trial and developing targets and so on, and then together with health economists, the interpretation of the trial. And we're going to try and use this individual-based simulation, which really integrates all of the data. I'm also leading the phylogenetics part of the trial. Phylogenetics is clearly useful. It's clearly very important to understand where transmission is occurring. These interventions are not going to eliminate HIV transmission. It's very important to know, what are the patterns of residual transmission in the intervention communities, and how do they differ from the control communities. You wouldn't believe quite how controversial phylogenetics has been in the context of such a trial, ethically and so on. There are 3 years of discussion over ethics, and the phylogenetics, despite the funders being very keen from the outset, as you might expect, and the phylogenetics component of the trial is going ahead in Zambia, but not in South Africa, and involves 20 further staff who are going around consenting patients for phylogenetics and collecting bloods and collecting the data and linking it to the clinical data. And it's an interesting exercise. A lot of phylogenetics work is done sort of post hoc, and you formulate the questions as you go, as it were, to actually write down from the outset what your aims are, and to prove without having done any work that you're going to be able to answer those aims is something which is pretty challenging. But building on the work of, for example, Eric Volz and Ollie Ratman studies and so on, and several other studies, we stated that the primary aims of this study were to estimate the role of acute infection, estimate the predictors of being an infector, and estimate the amount of transmission which is occurring from outside of the community, which is clearly very important for community-targeted trials. And secondary aims are to estimate the trends in antiviral resistance, to validate coalescent phylogenetic methods, and to contribute to the Pangaea HIV consortium. One thing which I don't think I'd seen, I'd love it if somebody says that there are methods for this, was actually how do you do power calculations for a phylogenetic analysis. So we ended up doing something that was quite simple, and Eric Volz confirmed that he got very similar estimates using a more complicated approach, is saying how many pairwise, how many recent coalescences do you expect for a given incidence and a given coverage? And for doing that, we showed that in order, for example, to tell the difference in between 40% of transmissions occurring in acute infection and 10%, we needed to sample at least 20 to 25% of the population. I'm sure with better methods, you could pull these confidence intervals back. But that resulted in us, because these were the questions we really wanted to answer, that resulted in this being a very large study. So just going right back to the start, really, how can we achieve these new goals, which have just been set by 2020 or by 2030? Can we actually show what will reduce HIV incidence? And phylogenetics surely has a very important part in that. And I think it's important to see phylogenetics as part of the standard public health toolkit. And I hope I've given you some flavor of questions from the point of view of public health. And obviously, we've seen many talks of people being much more directly involved in public health than we have. But just this whole, I think one of the things which I haven't seen and that we've really learned a lot from is by combining clinical, epidemiological, and sequence data. That does raise questions of privacy and so on. But I think there you can really get value. So just the outlook is that viral phylogenies resolve transmission, and they really should be seen as a key epidemiological tool along with the others. Trials now can't operate without mathematical modelers, it sort of says, and maybe they shouldn't be able to operate without phylogeneticists. I think maturation of this approach will involve integration into epidemiological toolkit, means that things like focus on study design and integration of other powerful approaches, such as household studies, cohort studies, trials modeling, how do you actually incorporate those into combined estimates? And then what can phylogenetics, apart from the insight, bring? There isn't this culture of open data sharing and community science in much of epidemiology. And as the field integrates more into epidemiology and public health for those who want to go in that direction, I think it's very important to stand by your ground, our ground, in terms of that community of open data. I would say your ground, being a more relatively recent entrant into this. I think that culture would be, everything I've seen at this conference in particular, but elsewhere, shows that culture would be a huge bonus to the field. And it's not something that should get lost, but actually spread as a result of that integration. Thank you. And thanks most of all to all of my collaborators and funders.
Thank you. So you mentioned briefly about recombination making it impossible or hard to resolve more basal lineages and elements of the phylogeny. I wonder to what extent, so in principle, yeah, I guess my question is, can you see if, in cases where there's a lot of recombination, can you somehow detect that and can you post-process your phylogenies so that you know if, because we know that recombination can potentially mislead inference quite a lot. Can you detect that, and can you guard against that? Also in the case of more recent lineages, or generally speaking.
So it's a great question, and I think you probably can for recent lineages, and it's there are methods out there for detecting recombination on consensus genomes, and you can apply those. I think those are very good at resolving recombination between quite distant lineages, but not the sort of intrasubtypic lineages. I think that with the summary statistic approach that we're using, we hope to be able to resolve those recent recombinations, so that when we see, a consistent signal of the virus being in one part of the phylogeny, we're working with Caroline Kalein's group in sort of characterizing all of these different trees in one go. When you see the trees, all of the tips are one part of the tree for this bit of the genome, and they're all in that part of the tree in another part of the genome, then you can reconstruct those haplotypes and phylotypes, we call them, and then from those you can resolve the phylogeny recently. To go deeper back is, on the other hand, because deeper back into the phylogeny, these become splits and so on. And there are these approaches, for example, based on the grand recombination coalescent and so on. I know that I've seen some papers where people have started to do that. I would imagine that's a very challenging task. Doesn't mean it's impossible, but very difficult, I think.
Excellent talk, excellent, and really nice tools to really look at the epidemic. I was wondering, maybe we should look in all these court cases and see what you can learn from that for modeling the epidemic. I see a lot of court cases where it looks like gay men go to parties and they go to several parties, and then they might get infected from several sources and reinfect each other back and forth. Now, how would you take that into account in your modeling?
Yeah, the back and forth transmission is something that I've also been curious about for many years. Actually, I've been curious about it in the context of heritability and this issue that we were getting different estimates for long-term couples than we were for the population. Actually, now the estimates are all consistent. I'm less concerned about that. But I think the intensity of transmission and multiple exposures and so on is something that we need to understand and help identify core groups. One of the things that I think there's a lot of sociological and the court case evidence and so on really does confirm the importance of these sort of very, very high risk, short periods of time. And the fact that people are testing, very close to the points where they get infected. I mean, obviously people know when they're at risk. And I think, pre-exposure prophylaxis has a huge role to play in those sorts of settings. We know much less about the epidemic in Zambia and South Africa. There's lots of sociological evidence about different types of high-risk behavior. None of them, to my knowledge, has been conclusively associated with HIV transmission. I think alcohol use, drug use are very clearly associated with HIV transmission. And linking to that kind of data is important for understanding transmission. But it's always very hard to do this in a way which, it does touch on events which very easily result in people being stigmatized. So yes, it's the short answer. I think you're absolutely right. Understanding where and how transmission takes place is what this is all about. And cross-linking with other sources of information. One of the things we're doing, incidentally, is looking at these people who are dually infected across the Pangaea and asking, is there something different behaviorally or epidemiologically amongst people who are dually infected compared to people who are singly infected?
Yeah, very fascinating. So you discussed that you have this signal of sort of dual infection. Can you give us a sense how rapidly you lose it as you go deeper into infection, like after 6 months or a year and so forth? When do you stop being able to detect it?
No, I can't, because all of our individuals, because of the way the study was designed for the GWAS, they're all relatively recently infected, all within 6 to 24 months of infection. I think you have a dually infected patient that you followed for several years. So.
Early on, but not later. Right?
Okay.
But we don't have many examples.
No, I mean, it would be really, one of the things that I think would be really interesting would be to choose a subset of these patients and really go into longitudinal depth. But because we decided to maximize our power for the primary aim of the study, we decided one sample per patient initially.